Welcome back to All About RX. Today, we are breaking down the core principles of neural communication by exploring exactly how neurons generate electrical signals through membrane potentials. If you've ever wondered how a thought instantly travels through your brain, or how a signal tells your hand to move in literally a fraction of a second, well, you're in the right place. We're going to look closely at the microscopic domino effect that makes all of this happen.
To conquer this, we're following a quick roadmap. Resting and graded potentials, firing and action potential, voltage-gated ion channels, the refractory period, how neural signals travel, and finally, the anatomy of a neuron.
Let's just jump right into section 1, resting and graded potentials. We're starting with the calm before the storm.
So, every cell maintains a resting membrane potential. In neurons, this baseline is typically around -70 millivolts. Why so negative? Well, it's all due to the uneven distribution of ions, stuff like sodium and potassium, and the differential membrane permeability between the inside and outside of the cell. Think of it like a tightly coiled spring, electrically speaking, just waiting to snap. When we nudge that coiled spring, the potential shifts from its resting state. A new signal causes depolarization, meaning it gets less negative and moves up towards zero. Then we have repolarization, dropping right back to the baseline. And sometimes it overshoots into hyperpolarization, dipping even further below -70 before finally stabilizing.
Now, when a triggering stimulus opens gated channels, it produces local, short-distance changes known as graded potentials. It's kind of like dropping a pebble in a pond. The ripples are strongest at the center, but they spread decrementally and die out within a few millimeters. But if those local changes depolarize the membrane enough, they trigger something massive, an explosive action potential that follows the all-or-none law and travels long distances without ever fading.
Which brings us to section 2, firing and action potential. What actually happens when we hit that explosion point? If those graded potentials push the membrane to the critical threshold, which is usually between -50 and -55 millivolts, boom. An explosive action potential is fired. It shoots up in a rapid, roughly 100 millivolt spike, peaking around +30 millivolts in about 1 millisecond. It is a wildly fast biological event. Breaking that spike down, it happens in a really precise sequence. First is a slow depolarization to threshold. Second is that explosive rising phase to the peak. Third, a rapid repolarization, or falling phase, where it crashes back down. And fourth is that after hyperpolarization undershoot, before it finally resets. So what's physically driving these massive swings? That takes us to section 3, voltage-gated ion channels.
That roughly 100 millivolt spike involves the sequential opening of voltage-gated ion channels. First, hitting the threshold triggers a massive influx of sodium ions through voltage-gated sodium channels during the rising phase. Then, right at the peak, those channels slam shut, and voltage-gated potassium channels immediately open up for a massive potassium efflux during the falling phase.
This is the perfect example of the all-or-none law. It's exactly like firing a gun. The trigger is either pulled hard enough to reach that threshold and the neuron fires fully, or it's not and nothing happens. There's absolutely no partial firing. But after it fires, the neuron has to protect the signal's direction. Let's look at section 4, the refractory period. To ensure signals travel in only one direction, absolute and relative refractory periods completely lock down the membrane right behind the active wave. The area that just fired gets temporarily exhausted, and the sodium channels basically refuse to open. This brilliantly prevents backward reexcitation and forces the signal to travel strictly forward. Now that we know it only goes forward, let's explore exactly how it gets from point A to point B in section 5. How neural signals travel. Depending on the fiber type, the impulse propagates in one of two ways. In unmyelinated axons, we see contiguous conduction. It moves step by step, depolarizing every adjacent patch, like a stadium wave. But in myelinated fibers, which are wrapped by Schwann cells and oligodendrocytes, we get saltatory conduction. The impulse leaps rapidly node to node, completely bypassing the insulation to speed up communication significantly. It's roughly 50 times faster. Let's tie all this microscopic action together in section 6, anatomy of a neuron. Looking at the whole cell, those graded potentials happen in the branching dendrites and the cell body. The signal converges at the axon hillock, and if the threshold is met, the action potential speeds down the axon,
riding that fast-conducting myelin until it hits the axon terminals, releasing chemical messengers to talk to the next cell. So, just a super quick recap of the absolute core takeaways. We've got our resting potential around -70 millivolts, a threshold of -50 to -55. The rising phase is your sodium influx, the falling phase is your potassium efflux, and saltatory conduction is that incredibly fast node-to-node jumping. I'll leave you with this to think about. If saltatory conduction is so crucial for speed, what happens in conditions like multiple sclerosis, where the body actually attacks its own myelin sheaths? Well, the scarring drastically slows or blocks these vital electrical impulses, showing just how beautifully but fragilely our nervous system is engineered. That wraps up our deep dive into neural communication. If you found this helpful, make sure to hit that like button, subscribe to AllAboutRx for more physiology and pharmacology breakdowns, and drop your questions in the comments below. See you in the next explainer.